Welcome to the pathogens and disease section of Year 12 biology. It's important that we are able to clearly define what a pathogen is. It is a disease-causing organism. The pathogens that we cover in the WACE course are bacteria, viruses, fungi, and protists. Today, we'll be concentrating on the structural features of bacteria. However, before we begin to examine the structural characteristics of bacteria, we should think back to the five-kingdom system of classification. Bacteria belong to kingdom Monera. These are our prokaryotic organisms. Remember that prokaryote translates to before the nucleus. So bacteria do not have membrane-bound nuclei, and their genetic material simply floats freely around the cell. We can describe bacteria as microscopic unicellular prokaryotes. They have a cell wall made of carbohydrates and protein and DNA with no nuclear membrane. Apart from ribosomes, they have no other membrane-bound organelles. They have single circular chromosomes and often have flagella or pili present. Flagella can be recognized as a tail-like structure, while pili are hair-like structures that cover the surface of the bacterium. The singular of flagella would be flagellum, and for pili, the singular term would be pilus. Bacteria contain a single circular chromosome and plasmids. You may remember that we covered plasmids previously in the course when we spoke about producing recombinant DNA while covering transgenic organisms. Bacteria also have a neat trick of transferring their plasmids with other bacterial cells, and this can be a useful way of sharing beneficial alleles such as antibiotic resistance. While this is outside the scope of the course, please be aware that bacteria come in a wide range of shapes. Scientists classify bacteria based on their shape, and we can see a range of examples in the diagram. Bacteria reproduce asexually through binary fission. Here we see an outline of the stages of binary fission from a previous WACE examination. This indicates the level of depth that is required. So starting for the first step of binary fission, DNA is replicated, genetic material moves to opposite sides of the cell increases in size. A double wall develops across the midline of the enlarged cell. Each copy of DNA attaches to a different part of the cell membrane. The cell separates into two daughter cells at the midline wall. Remember that a defining feature of binary fission is that each daughter cell will have a copy of DNA which is identical to the original parent cell. Bacteria can reproduce extremely quickly, with a new generation occurring every 20 minutes or so. This feature provides the perfect conditions for any beneficial mutations to spread rapidly across a population. Bacteria can cause disease in three main ways. Number one, they can be invasive. They spread through tissues and can damage cells with their digestive enzymes. Number two, they can produce toxins that damage cells. An example of this could be tetanus. Number three, the host organism's own immune system might damage cells through high levels of inflammation. Antibiotics are used to treat bacterial infections. They work in two main ways: by either changing the cell wall or membrane, or by stopping replication or protein synthesis. Antibiotics can be broad spectrum and are able to treat a wide range of bacteria, or narrow spectrum, which are effective against only specific types of bacteria. This brings us to the end of our video and the introduction of the structural characteristics of bacterial cells. The three important pieces of information from this presentation are that bacteria are unicellular prokaryotic cells, they reproduce asexually using binary fission, and that bacterial infections can be treated using antibiotics. I hope you found this video useful. Happy studying!